Hi students, how are you all? I hope all of you are doing good. Okay, I am Geeta, botany faculty at Gosala Zone, Sri Chaitanya campus, and today I am going to give you an explanation about the meiosis, a chapter cell cycle. It is there in the botany, and in that I have already dealt with the mitosis before. Now let us see today meiosis. Meiosis is an important step of cell division which is happening in the reproductive cells, particularly in the reproductive cells. This will happen. So generally the meiosis will be happening in the reproductive cells and not in the somatic cells in majority of the organisms. And this meiosis is classified into different types based on where it is happening or what it results in. For example, if the meiosis is happening in the zygote, it is called as zygotic meiosis. If it involves or if it is happening in the zygote, it is called zygotic meiosis, which is present in the primitive organisms like algae and fungi. Algae and fungi. They show the zygotic meiosis where the meiosis will be happening in the zygote. The second type is sporic meiosis. Sporic meiosis, which occurs in the formation of spores. It occurs in the spore mother cells and it results in the formation of spores here, and this type will happen in the plants. In the total plants which you have learned, bryophytes, pteridophytes, gymnosperms and angiosperms. Then the last type is gametic meiosis. Gametic meiosis. This occurs in the formation of gametes. It occurs in the gamete mother cells. It results in the actually formation of gametes. So it is called gametogenesis. Formation of gametes, this occurs in the gamete mother cells. And this happens generally in the animals, animal kingdom. So there are basically three types of meiosis: zygotic meiosis, which occurs in algae and fungi, then sporic meiosis, which occurs in the formation of spores in plants, gametic meiosis, which occurs in the formation of gametes or gametogenesis that happens in the animals. Wherever it is associated with the generally reproductive cells. The somatic cells, if the term is given, then that cells will be undergoing the mitosis. Reproductive cells will be undergoing the meiosis, or these are also called as germ cells. So name the type of division which occurs in germ cells or reproductive cells. If they ask that, it is meiosis. And always the meiosis will happen in the diploid cells. Always it happens in the diploid cells. That is the nature of the meiosis. And let us see this meiosis in detail further. This is the basic information which happens in that. And the cells which are showing the meiosis are generally called as meiocytes. There is a term which is given for all these. They are called as meiocytes. And this meiosis, it has a nickname also. It is also called as reductional division. Reductional division, because it reduces the chromosome number exactly to half. So when we take a parent cell which is diploid in nature, it will be dividing into 4 daughter cells that are haploid by the end of the meiosis. That is the nature. So as it is reducing the chromosome number exactly to half, it has a nickname which is called as reductional division. And it will convert these diploid cells always into the haploid cells. It will form the daughter cells which are always haploid. And the number of daughter cells which are formed will be generally 4 in number. 4 daughter cells are formed at the end of the meiosis. Now let us see the further details of the meiosis, how it has been classified into further types. As you all can see, the meiosis is divided into meiosis 1 and meiosis 2. So here you have the meiosis, which is divided into meiosis 1 and meiosis 2. In meiosis 1, okay, you have the karyokinesis 1 and cytokinesis 1. These all 4 will come under. Karyokinesis 1. After this one cytokinesis will be there. So this is the first one, this is the second one, but these two are happening in the meiosis 1. And then in meiosis 2, we have again these 4 will be coming under karyokinesis 2, and after this we have cytokinesis 2. So meiosis is actually divided into 2 phases, meiosis 1 and meiosis 2. Meiosis 1, it includes one time nuclear division, karyokinesis 1, and followed by one time cytoplasmic division, cytokinesis 1. Then meiosis 2 is further subdivided into karyokinesis 2 and cytokinesis 2. But in between here, in the middle of these two, interkinesis will happen, interkinesis. So between meiosis 1 and meiosis 2, interkinesis occurs where there is no DNA replication. It will not be involving the DNA replication at all. Just the cell will be preparing itself to undergo the meiosis 2. That's all. No much changes will be there. Just RNA, proteins will be synthesized. No DNA replication will happen in the interkinesis. So basically meiosis is divided into 2 phases, meiosis 1 and meiosis 2, with in between interkinesis. And meiosis 1 is further subdivided into the karyokinesis 1, where first time nuclear division will happen and followed by cytokinesis 1, cytoplasmic division, and then meiosis 2 is again subdivided into karyokinesis 2 and cytokinesis 2. This is the second time nuclear division. So we can conclude saying that meiosis has 2 times karyokinesis or 2 times nuclear divisions and 2 times cytoplasmic division. 2 nuclear divisions. It includes 2 nuclear divisions and it includes 2 times cytoplasmic divisions. These are happening in the sequence. First one nuclear division, after that it is followed by cytokinesis, cytoplasmic division, then again one more time nuclear division, and that is also followed by the second time cytoplasmic division. So it includes basically 2 nuclear divisions, 2 karyokinesis and 2 cytoplasmic divisions, 2 cytokinesis 1 and 2. And in between them we have the interkinesis. This is the basic information of the meiosis. Now let us see the textbook diagrams which are given in NCERT. In NCERT, the meiosis 1 stages are represented in these diagrams, and if any diagram based questions come, you have to identify these pictures. So that is the reason why I have taken the pictures mainly from the NCERT. The first picture shows that it is prophase 1. It is prophase 1. And in this prophase 1, all of you can see very clearly that red color, somewhat red color chromosomes are there, and even blue color chromosomes are there. The total number of chromosomes you can see 4 are there. 4 chromosomes are present, okay, in the prophase 1. So if they give like this, okay, they are showing the intertwining or they are connected at some points. If they give this diagram, it is the prophase 1. And metaphase 1, you can see that chromosomes, red ones and blue ones, they are placed on one line, and this down one also, one more line is there, they are aligned on the equator. This is the metaphase 1. In anaphase 1, this red chromosome and blue chromosome, complete chromosomes, they are moving towards the opposite poles. These two are moving towards this and these 2 chromosomes are moving towards this. This picture is anaphase 1. Then telophase 1, the end of that, these 2 chromosomes are developing a nuclear envelope and it becomes one daughter nucleus here, and this one more daughter nucleus. These 2 are called dyads of cells. 2 daughter cells are formed, okay, once the cytokinesis is completed. Before cytokinesis, these 2 are now called as 2 daughter nuclei. Daughter nuclei, they are called as. Once the cytokinesis also is completed, then these 2 are called as dyads of cells. So this is a picture which is given in NCERT, and you have to identify which stage is what. The first picture is prophase 1, where the chromosomes are elongated thread-like and they are intertwined among themselves. And in metaphase 1, they are aligned on the equator. This, if you see, it is metaphase 1. If they are getting separated, if they are moving towards the opposite poles, it is anaphase 1. And 2 daughter nuclei, if they are formed properly, that is the end of the telophase 1. Then you can see here the same picture, it is given in the NCERT, and this is the stages of meiosis 2. In the meiosis 2, prophase 2 is the first phase, and in that blue one and red one is taken from the previous diagram, and the blue one at the end of the telophase you can see clearly here in this. We have got one red chromosome and one blue chromosome here, and we are continuing now with this. One red and one blue chromosome in one cell. This is one cell, or you can consider this as a daughter nucleus. Daughter nuclei. So 2 daughter nuclei are there, 1 and 2, and they are all having, these 2 are having red one and blue one here. So if this picture is given, prophase 2, and that leads to the metaphase 2, where this red and blue ones are arranged on the equator, and if they are getting separated, it is anaphase 2, and by the end of the telophase 2, you are getting 4 daughter cells. As I said, 4 daughter cells are formed, and all these are having, if you see the before diagram, you can see that the number of chromosomes here are totally 4, and now you see here at the end of the telophase 2 and cytokinesis 2, in each cell we have 1 and 2 chromosomes. So initially the parent cell was having the 4 chromosomes, but in the daughter cells at the end only 2 chromosomes are present. So this is the reason why 4 chromosomes they have reduced into 2 chromosomes. So this meiosis is actually called as reductional division, where the chromosome number is reduced to exactly half. Then let us see the subphases of meiosis 1. Okay. So in the meiosis 1, we have the karyokinesis 1 and cytokinesis 1. In karyokinesis 1, we have the 4 phases: prophase 1, metaphase 1, anaphase 1 and telophase 1. But among this meiosis 1 and meiosis 2, much more complex phase is the meiosis 1, and in that especially the complicated phase is the prophase 1. This is the more complex phase, and it is subdivided into 5 subphases. The 5 subphases are leptotene, zygotene in the sequence, pachytene, diplotene and diakinesis. So there are basically in the prophase 1 we have 5 subphases. 5 subphases are there, and they are named as leptotene, zygotene, pachytene, diplotene and diakinesis. In the leptotene, all of you can see clearly in this. The first phase is the leptotene. This is prophase 1. Subphases. So in the leptotene, all of you can see that the chromosomes are present like elongated threads. It is chromatin material only, and they are present in the form of elongated threads, which are having some bead-like units. Some beads are present here, and these beads are called as chromomeres. Chromomeres. So all the chromosomes are distinctly visible. They started undergoing the condensation, and they started distinctly visible with elongated threads with beads on them, and that beads are called chromomeres. And all these chromosomes, okay, they will be attached to one side of the nuclear membrane, which looks like somewhat bouquet of flowers. So this is also called bouquet stage. So in the leptotene, the chromosome starts appearing distinctly visible. That is the first stage of the prophase 1 leptotene with the chromomeres, the bead-like structures. Then it enters, the cell enters into the zygotene. In the prophase 1, you are learning about the karyokinesis. In that, leptotene is the first one, where chromosomes are distinctly visible with chromomeres. Next, the cell will enter into the zygotene. In the zygote, what happens? The two chromosomes which are exactly alike, similar, okay, like in terms of structure, in terms of size, in terms of genes, in terms of gene arrangement, in terms of gene numbers, if everything is same, then the two types of chromosomes are called as homologous chromosomes. Homologous chromosomes. The two chromosomes which are identical in all aspects, they are called as homologous chromosomes. So this type of homologous chromosomes will be there inside the nucleus in different places. So in the zygote, what happens, that homologous chromosomes, wherever they are inside the nucleus, they will come towards each other because of some attractive force between the two homologous chromosomes. That attractive force is due to particular proteins between them. Some attractive force will be developed between them. These proteins are called as synaptinemal complex proteins. Synaptinemal complex proteins. So some complex proteins will be there. They will be attracting the homologous chromosomes towards each other. And because of this synaptinemal complex proteins, the two chromosomes which were far here in the leptotin, they will try to come towards each other. That is the synaptinemal complex. So the attractive force between the homologous chromosomes is done by the synaptinemal complex. And once the synaptinemal complex brings them together, then the resultant structure, okay, now which has two homologous chromosomes together, the two homologous chromosomes are together, it is called as one bivalent. So you can see here clearly two chromosomes are pairing up here, and the two chromosomes which are pairing up here, okay, they are now called as bivalent. Each pair is called as a bivalent. So by the end of the zygote, bivalents are formed by the combination of two homologous chromosomes, or coming together of two homologous chromosomes together. The process of pairing which happens in the zygote, it is called as synapsis. So synapsis is nothing but the process of pairing of two homologous chromosomes which occurs in the zygote stage, and the end result of the zygote, inside the cell nucleus, all bivalents will be present. If the parent cell is having 46 chromosomes, the bivalents which are formed here will be 23 bivalents, because one pair is equal to one bivalent. So like that we have 23 pairs, so 23 bivalents will be formed in human cells. So by the end of the zygote, bivalents are formed by the process of pairing, that is called synapsis, and it is happening by the synaptinemal complex proteins. Now this type of cell nucleus enters into the next stage, the third stage, that is called as pachytene. This is the second stage, zygote, and third stage is the pachytene. In pachytene, what happens is each chromosome, okay, will be further condensed, because condensation started in the leptotin. You can see here they are elongated thread like, here condensation is continued, they are becoming short. Condensation started already in the leptotin and they are becoming condensed. Here each chromosome will appear to have two sister chromatids that are attached at the centromere. Each chromosome will be having actually two sister chromatids from the beginning after S phase. In S phase, the single chromatid containing chromosome replicated, and each chromosome from S phase onwards, it will be having two chromatids. But due to lack of condensation, here we can't see two chromatids, two sister chromatids we can't see, they are not visible, but they are present. In zygote, they started undergoing condensation. In pachytene, much more condensation happened. So in this we can see very clearly each chromosome will be having two chromatids like this. The two sister chromatids will be there like this, and they will be joined at the centromere. And now that sister chromatids which are joined at the centromere, they are present for the bivalent. You know that one bivalent will be having two homologous chromosomes, and now each bivalent will be having four sister chromatids. So one bivalent is equal to four sister chromatids. Four chromatids we can write actually, because chromatids which belong to one chromosome are called as sister chromatids, and chromatids which belong to two different chromatids are called as non-sister chromatids. So now in the Paketine stage, due to the further condensation, each bivalent will possess four chromatids. So now each chromosome pair or bivalent will be looking like four chromatids. So it is called as tetrad. As it is having four chromatids, it is only called as tetrad. One tetrad. These are all same. Two homologous chromosomes is equal to one bivalent is equal to four chromatids. Four chromatids containing structure is called one tetrad. But you can see the tetrads only in the Paketine. So they are also called as Paketine tetrads. Tetrad is nothing but having four chromatids in the bivalent or in the two homologous chromosomes. So in the Paketine, due to condensation, the tetrads are formed. Okay, and in all the subphases of prophase 1, this is the much complicated phase and it takes longer time. Though the prophase 1 is the longest phase of the meiosis, in this the Paketine will take lot of time particularly, and it is a more complex stage because these two are smaller stages, because they will take less time. But compared to the first two, this is taking more time. And here Paketine, after the condensation, okay, we can see the four chromatids containing structure very clearly, and that each structure with four chromatids, it is called as one tetrad. So this stage is called as tetrad stage, Paketine tetrad stage. And in this stage, what happens is, each chromosome which is having four chromatids here like this, okay. In the tetrad we have the four chromatids. So the pink ones, the pink two chromatids are called as sister chromatids because they belong to one chromosome, and the white ones, they belong to one chromosome, so they are called as sister chromatids. Take one pink and one white. These two you take, these two are called as non-sister chromatids. So these two non-sister chromatids, okay, they will be forming recombination nodules. You can see here, this is given. Between the non-sister chromatids, recombination nodules are formed. Recombination nodules are formed. So two non-sister chromatids of one bivalent, they start, okay, pairing up each other, or they will be exchanging their materials, okay. One break will be happening and they will join to the other end. Like that, it always happens between the two non-sister chromatids. And the process of exchange of genetic material between the two non-sister chromatids of a bivalent, it is called as crossing over. It is called as crossing over. So in the Paketine stage, crossing over occurs. This is the most important phase of the meiosis 1, because the exchange of genetic material happens between the two non-sister chromatids of a bivalent, okay, with the help of a particular enzyme. The enzyme is called as recombinase. Recombinase. So with the help of that recombinase, okay, one non-sister chromatid and another non-sister chromatid, they will break their parts, and this part will come to this and this part will come to this. The red one will go to the blue one and blue one will come to the red one. So the process of exchange of genetic material, which is called as crossing over, that occurs in the Paketine stage. It happens in the Paketine stage. Now this type of nucleus will enter into the diplotin stage. In diplotin, what happens is, all of you can see clearly in this, chiasma is given. Chiasma singular, chiasmata plural. So here synaptinimal complex proteins were there in the zygote onwards. Now in the diplotin, what happens is dissolution of the synaptinimal complex will happen. Whatever the proteins were there that were holding these two chromosomes together, that will be dissolved. So the attractive force between these two chromosomes of a bivalent will be dissolved. Dissolution of synaptinimal complex proteins occurs here, and that leads to the separation of homologous chromosomes of each bivalent. This chromosome will try to separate this way and this chromosome try to separate this way. They will start separating away from each other due to the dissolution of synaptinimal complex proteins. Here when they are separating, still in the packaging they are linked at the points of the crossing over. So when they are trying to separate, you can see they will form one kind of X-shaped structures. X-shaped structures you can see very clearly. This is the X-shaped structure which is formed, and these X-shaped structures are called as chiasmata. They indicate the sites or points of crossing over. As the homologous chromosomes of a bivalent are getting separated due to the dissolution of the synaptinimal complex proteins, we can clearly see the X-shaped structures called as chiasmata in diplotin stage. So the X-shaped structures are clearly seen in the diplotin. And in fact, than packaging, diplotin is lasting for so many months or years in some of the organisms like oocytes of vertebrates. In some of the oocytes of the vertebrate amphibians, particularly secondary oocytes and all, this stage they will be arrested. Okay, other than that, in the remaining organisms, we can think that packaging is taking time longer. If you take the oocyte of vertebrates, diplotin stage lasts for months or years. It can be there for months or years. So in the diplotin, condensation happened and dissolution of the synaptinimal complex proteins occur, and the two chromosomes of a bivalent, they try to separate or away from each other. That results in the visible structures of X-shaped structures called chiasmata. They are seen here, but they are formed in the packaging. They are the points of crossing over, points of exchange of genetic material. That X-shaped structures are called chiasmata. Chiasmata are very clearly seen in the diplotin. Then after the diplotin, the cell enters into the diakinesis. In the diakinesis, what happens? The chiasmata formation from this point, it will reach, from this point, it would have reached to the ends of the chromosomes. The chiasmata will try to separate the point of origin to the ends of the chromosomes like this. So the movement of chiasmata from the origin point to the ends of the chromosomes, it is called as terminalization. Terminalization. So the chiasmata is formed here, and from this point of origin to the ends of the chromosomes, it is moving because they are getting separated away from each other. So the terminalization of chiasmata occurs in the diakinesis. So by the end of the diakinesis, the chiasmata will move towards the ends of the chromosomes, but still the chromosomes are connected by their ends. And at this stage, centrioles would be migrating towards the opposite poles and complete condensation happened, and this nuclear envelope breaks here, okay, or disappears. Nucleolus disappears, Golgi complex ER disappears. The events of prophase of mitosis will happen here. And now all the chromosomes are released into the cytoplasm, and meiotic spindle is also formed, and that spindle will be connected to the chromosomes which are already there connected at their ends. That is the end of the diakinesis. Now the cell from this diakinesis will enter into the metaphase 1. In the metaphase 1, All these bivalents which are attached at the ends, they will be connected to the respective pole by one spindle fiber. See here, this one chromosome is connected to one spindle fiber, one pole, and this chromosome is connected to another pole. So a bivalent is connected by two spindle fibers, okay, and one chromosome is connected by one spindle fiber from each pole. So that in the metaphase 1, what happens is, by the contraction and relaxation of all these spindle fibers, okay, they will be bringing all the homologous chromosomes which are attached at their ends to the equator of this cell to form the metaphase plate. So all the chromosomes of that cell, which are like pairs now, that pairs will be brought to the center of this cell to form the metaphase plate or equator. Okay, that is forming the metaphase plate. So briefly you can see here the prophase 1 subphases: leptotene, zygotene, pachytene, diplotene and diakinesis. This is the longest phase. So which is the longest phase of meiosis 1? It is the prophase 1 because it has the 5 subphases. Okay. And leptotene, the chromosomes are seen like elongated threads. Zygotene, pairs you can see. The bivalents are formed here. Chromosome pairing starts. The process of pairing is synapses and each results in the bivalent formation. Now in pachytene, sister chromatids are visible for each chromosome and each bivalent is having 4 chromatids each. So these are called tetrads. And that pachytene tetrads, they exchange their genetic material after forming the recombination nodules with the help of recombinase enzyme, and they exchange their chromosomal material or genetic material between the red and blue one. Okay, and the process of crossing over happens here. The exchange of genetic material is nothing but crossing over. It happens in the pachytene. Then in diplotene, as the synaptinimal complex dissolves, they try to move away from each other. And at that time, X-shaped structures called chiasmata are very clearly seen because further condensation happened. And then this is the longest phase in the oocytes of vertebrates, lasts for months or years. In diakinesis, terminalization of chiasmata will happen. The chiasmata from origin point, it will move towards the ends of the chromosomes, terminalization of chiasmata. Then all these chromosomes will be released into the cytoplasm. In the metaphase 1, you can see in the metaphase 1 here. In the metaphase 1, all the chromosomes, bivalents, are placed on the equator. The bivalents are placed on the equator of the cell to form the metaphase plate. In anaphase 1, the sister chromatids containing one chromosome, bivalents get separated. That means a complete chromosome with 2 sister chromatids, a complete chromosome will be moving towards one pole with 2 sister chromatids. So bivalent separates in the anaphase 1. There is no centromere splitting in anaphase 1. Just the homologous chromosomes which were together before, they get segregated towards the opposite poles. And this is separation of bivalents or separation of homologous chromosomes completely, okay, with their sister chromatids. Both the chromatids will be there for that, no centromere splitting, and they will move towards the opposite poles in the anaphase 1. Later, the cell will be entering into the telophase 1, where the chromosomes which have reached the opposite poles, okay, the reverse of the prophase 1 will happen. Nuclear envelope reappears. Nucleolus reappears, Golgi complex ER will be developed, and these chromosomes actually they will go to the extended form, but not like the fully extended form of the interphase. They will show some kind of dispersion, they will expand, but they will not reach the size of the interphase chromosomes, interphase chromatin. They will show only medium size extension. And after that, 2 daughter nuclei are formed at the end of the telophase 1. Now the cytokinesis will happen and it will form, the cytoplasm will be dividing into 2 and it results in the formation of 2 daughter cells, and these 2 daughter cells are called as dyads of cells, which are formed by the end of the cytokinesis 1. So, in the meiosis 1 particularly, one cell, one diploid cell is getting converted into two daughter cells which are haploid. Two haploid cells are formed. Two daughter cells are formed, so they are haploid and they are called as dyads of cells. Now, these two dyads of cells will enter into the meiosis 2. Those two will enter into the meiosis 2. In the meiosis 2, what happens? You know, in meiosis 1 and meiosis 2, in between we have intrakinesis, where the DNA will not replicate, but the RNA protein synthesis will happen, and before the chromosomes are fully extended up to this much expansions only, the cell will enter into the prophase 2. That means chromosomes are not reaching the fully extended state, they will reach only this much medium size extension, and immediately after that the cell will enter into the prophase 2, where the chromatin which got extended this much, it will start undergoing the condensation further, and again chromosomes are formed. Now you can see clearly in this, red one has the blue ends, and blue one has the red ones. That means already they exchanged their genetic material. Now the chromosomes are formed in this cell. So in the prophase 2, a new spindle forms around the chromosomes, and all the events of the prophase of mitosis will happen here. Meiosis 2 is actually called as meiosis 2 is equal to mitosis, because all the phases of meiosis 2 will be similar to mitosis. Prophase 2 is similar to prophase. This is similar to, the events are similar to prophase of mitosis. These events are similar to the metaphase of mitosis. These events are similar to the anaphase of mitosis. These events are similar to the telophase of mitosis. All the events are same. But the only difference is the chromosomes would have already exchanged their genetic material. So they will be having other chromosome parts. Red one has the blue one has the red one. Just the crossing over already happened, and they are different chromosomes now. They have different genetic material. So prophase 2 is equal to prophase, and where the chromosomes will be condensed fully, and then nucleolus, nuclear envelope disappears, and the chromosomes are released into the cytoplasm, and spindle is formed here. Then the cell will enter into the metaphase 2, which is like metaphase of mitosis, and all the chromosomes, okay, they will be aligned on the equatorial plate in each daughter cell. We have two daughter cells, dyads of cells. In each daughter cell, this is going to happen. So all the chromosomes are attached to the spindle fibers, okay. Now each chromosome is attached to the two spindle fibers from both the poles, like the metaphase of mitosis. And now they will form the metaphase plate in each cell, and two cells are there, two metaphase plates will be formed. In anaphase 2, here centromeres divide. They exactly in the middle, they will split here, exactly in the center like this, and centromere will divide, and the sister chromatids will separate. The two sister chromatids which are held together by the centromere, they separate, and they will try to move towards the opposite poles. This part is coming here, this part is coming here. You can see the color differences and all. So splitting of centromere happens in anaphase of mitosis and anaphase 2 of meiosis. And the daughter chromosomes are formed. Each chromosome will be having one sister chromatid, and they will try to move away from each other. And then in the telophase 2, the cell will enter into the telophase 2, where all the events of prophase 2 will be reversed. Here the chromosomes will reach the fully extended state, and they will become the chromatin. And already they have got the new genetic material, they have exchanged their parts. Now nucleolus will be formed, nuclear envelope will be formed, Golgi complex and ER reappears, and it divides that single nucleus into two daughter nuclei. Now after cytokinesis, it results in the formation of two daughter cells like this. One daughter cell will be forming the two daughter cells. This is their second daughter cells, and here we have got two more daughter cells. This is 1, 2, 3 and 4. So by the end of the telophase 2 and cytokinesis 2, we will be getting 4 daughter cells with half the number of chromosomes, exactly half the number of chromosomes. So here, whatever the cell you take in the meiosis, take a diploid cell, okay, in the meiosis. This is called as parent cell or meiocyte. And by meiosis 1, by the end of meiosis 1, it will divide into two daughter cells which are haploid in nature. These two are daughter cells. So diploid cell, the parent cell, will split up into two daughter haploid cells. These are called as dyads of cells, according to NCERT. Two daughter cells are formed which are haploid by the end of meiosis 1. Then these two daughter cells will enter into the meiosis 2. Okay, and they result in the formation of 4 daughter cells which are haploid. So by the end of meiosis 2, 4 daughter cells are formed from one diploid cell. This is how the meiosis will happen by involving meiosis 1 and meiosis 2. As it is reducing the chromosome number, 2n is becoming n by the end of this, but actually it happens in the end of the meiosis 1. The meiosis 1 is called reductional division. If they ask specifically, then you can tell this meiosis 1 is reductional division, or else this is the name for meiosis altogether. And meiosis 2, it is equal to mitosis, or you can call it as equational division. So this is how the meiosis will be happening, which involves meiosis 1 and meiosis 2, and one parent cell will be forming ultimately 4 daughter cells which are haploid in number of chromosomes. So this results in the end products of 4 daughter cells which are haploid in nature. Okay. Next, so we have completed the meiosis. As you have seen already in the diagram, you can see the meiosis 1, where you have prophase 1, metaphase 1, anaphase, telophase 1 and cytokinesis 1. Meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinesis 2. So in the meiosis, total meiosis, prophase 1 is the longest phase, and it is much complicated because it is divided into 5 subphases: leptotene, zygotene, pachytene, diplotene and diakinesis. Then after that, the metaphase 1, anaphase 1, telophase 1 and cytokinesis 1, it results in the formation of 2 daughter cells which are haploid in nature, dyad of cells. So meiosis 1 is actually reductional division because it reduces the chromosome number to half. Then this type will enter into the meiosis 2 after interkinesis. In between we have the interkinesis where no DNA replication happens, but RNA and protein synthesis will happen. Then the cell will enter into the 2 daughter cells are there. So each cell will enter, one daughter cell and second daughter cell will enter into the meiosis 2. Each daughter cell will undergo the prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis 2, and it forms 2 daughter cells at the end. As we have got 2 daughter cells, the 2 daughter cells will form 4 daughter cells at the end of the cytokinesis 2. So the parent cell, we have taken the diploid cell, and we have taken here 4 chromosomes. In the initial 4 chromosomes are there, blue one and green one, and in the daughter cells we got only blue and green one, 2 chromosomes are there here. By the end of this you have got 2 chromosomes, but only with one chromatid. Only one chromatid each chromosome is having. So in this way the meiosis will result in the formation of 4 daughter cells, as I have shown in this picture. It will be happening here. And they will ask the question, what about the chromatids here? Here the chromosome will be having this type of structure with 2 sister chromatids. I am taking for your understanding 2 chromosomes here. Now in this stage, the cells will be having, out of 2, 1 will be given to this cell and 1 will be given to this cell with sister chromatids. It will have 2 chromatids each. Now in this stage, each cell will be having the chromosome like this with only one sister chromatid. The amount of DNA is also reduced. Okay, if you have here 2 chromosomes, okay, 4C DNA if you take, here you are having the 2C DNA in each cell and here it will be having only 1C DNA. If you take in terms of chromatids, okay, here you will be having 4 chromatids, 1 bivalent will be there, here 2 chromatids, 1 chromosome, and here it will be having 1 chromatid and 1C DNA. In terms of DNA, if you write here it is 4C, 1C, 2C, 3C, 4C, then here in the cell, after the end of meiosis 1, you will be having the 2C and number of chromosomes is reduced to half. And by the end of the meiosis 2, each chromosome will be having only 1C amount of DNA. So the DNA has become, okay, 1/4 by the end of the meiosis 2. Number of chromosomes, it became 2 are there here, here only 1 is there. So 2 is becoming 1, so the chromosomes are reduced to half. So chromosomes means they are reduced to half. DNA amount if you take, here in this parent cell we have the 4C amount of DNA if you take, then by the end of the meiosis you will have 1C amount of DNA. Okay, this is the end of the meiosis, how it happens. Now the last point is significance of meiosis. What is the significance of meiosis? You already know that this meiosis will happen in the reproductive cells or germ cells. It is involved in the gamete formation and it reduces the chromosome number to half. Though it is reducing the chromosome number to half, it is involved in the conservation of chromosome number. It is involved in the conservation of chromosome number because it is exactly opposite to the fertilization. Number of a species. Okay, it is going to maintain or conserve the constant chromosome number in a species because it is exactly reducing diploid cells into haploid gametes, and then the haploid gametes will be undergoing the fertilization to form the zygote. So 2 gametes, imagine the male gamete is there and the female gamete is there, these are gametes. And the gametes are haploid, which are formed by the end of the meiosis, and these 2 will undergo fertilization and it forms a diploid zygote. And now these 2 cells are actually formed from the diploid cell only, parent cell, germ cell or parent cell, by the meiosis. So here we can say that by meiosis a diploid cell is being converted into haploid cells. By the fertilization 2 haploid cells are fusing to form the diploid cell. So meiosis is exactly opposite to the fertilization. The chromosome number, if it is not reduced by the meiosis, the formed product by the end of the fertilization would be polyploid. So meiosis is going to conserve a constant chromosome number in a species or of a species. This is the first significant point about the meiosis. And as you are all aware of, one process called as crossing over. Crossing over happens in the Paketine stage. They exchange their genetic material and results in the formation of different types of daughter cells at the end. Crossing over leads to recombinations. Recombinations leads to variations, differences among the organisms. These are important for evolution. So crossing over which happens in the Paketine, okay, that leads to recombinations of genes. That leads to variations and origin of a species. It is important sources for the evolution. This is the second point which leads to the variations, variability, or you can call it as genetic variability. Genetic variability, which are the important source of evolution. These two are the major significant points about the meiosis. That's all for the meiosis which happens in the reproductive cells. Thank you.